Alright, I actually realized that ideally we should open it with a different app. So I'm gonna start talking about it, and then, we'll okay, so kidneys, are they retroperitoneal? They are, right? Which kidney is higher? So, so left, because you have liver on the right side. Which blood vessels? Renal arteries, renal veins. So where do those blood vessels originate from? Aorta. And blood vessels drain the blood. Renal veins drain the blood into the vena cava. What's the point of entrance? Okay, that's hilum. Okay. Urine from the kidneys. Let's just kick a lot of things out right away. So there are two layers in the kidneys, right? There's a cortex and medulla. Cortex is superficial, medulla is deep. Microscopic structural elements of the cortex are renal columns. Medulla are renal pyramids, right? I'm gonna come back to this one a little later. What I wanted to say, that renal medulla, that's where the urine is produced, right? And urine drains from urine papilla into the minor calyces, then goes into the major calyces, then goes into the renal pelvis, then goes into the ureters, then urinary bladder, then urethra. Know this pathway, period. I'm going to ask about it. Okay? We got this. So we got this urinary bladder contained. How does a urinary bladder? It leaves through the urethra. Urinary bladder is anterior to rectum and to the uterus. 3 layers of connective tissue that surround kidney. That's renal fascia, perirenal fat tissue, and connective tissue capsule. Level of which vertebrae? L2. Renal pyramids. We got this just out, okay? So this is the pathway I told you about. Know this pathway. I promise there will be a question about it. The path of kidney flow. Main functional unit of the kidney, nephron. Know the parts. Glomerulus means filtration. Proximal, distal convoluted loops, and nephron loop are reabsorption and secretion. Okay? I will not trick you over with the questions like, where does most filtration, reabsorption occur? Okay, so reabsorption and secretion, it's all the tubular system. But know for sure that filtration is in the glomerulus. Glomerulus is filtration and nothing else. We're clear? Okay? From the distal convoluted tube, where does filtrate go? Collecting ducts. Okay? 3 main steps: filtration, reabsorption, and secretion. Nothing more than that. Okay? Please, don't create new steps. Right? Okay, we got this. So, clearly understand, filtration is everything goes from the blood to the filtrate. Everything except proteins and anything bigger than the protein. I'm sorry, which cells are responsible for the filtration? Podocytes. Filtration occurs because small molecules can go between filtration slits, and large molecules cannot. Okay? Type of capillaries in the kidneys is fenestrated capillaries. Reabsorption is moving of chemicals back into the blood, and secretion again from the blood into the filtrate. Know these directions. Are we clear? Two types of nephrons. Know the types and the function. So, cortical nephron is located where? Surprisingly, in the cortex. It's surrounded by peritubular capillaries. Peritubular capillaries, where do they receive blood from? Know the pathway for blood. Listen. So, afferent arterioles bring blood into the glomerulus. Efferent arterioles drain the blood from the glomerulus into the, for instance, peritubular capillaries. Blood from peritubular capillaries ends up in the veins. Okay? Are we clear about it? The main function of peritubular capillaries is acid-base regulation, electrolyte regulation, stuff like that. Second type is juxtamedullary nephron, which nephron loop is located, pretty much the entire thing is in the medulla, and the system of capillaries that surround the nephron loop of juxtamedullary nephron is vasa recta. Nephron loop of juxtamedullary nephron serves as the countercurrent multiplier. I don't think I have ever said that many polysyllabic words in one sentence. So that long loop is countercurrent multiplier. Vasa recta, capillaries around it, is countercurrent exchanger. The main responsibility is water balance. Okay. What else? Again, pathway. Afferent arteriole glomerulus, efferent arteriole, then either peritubular capillaries or vasa recta, and then vein or venule, it doesn't really matter. Does that make sense? Have it here. Okay? Glomerulus. Know that it's a capillary surrounded by podocytes, and all of it is in the capsule. Okay? Afferent arterioles are wider, and efferent are narrower. That's why blood pressure is high in the glomerulus. Okay? Afferent arterioles deliver the blood there, efferent drain it. Does that make sense? Three pressures that form net filtration pressure. We have two hydrostatic pressures. One is capillary, and that is basically blood pressure. Another is capsular, and that is a pressure of a filtrate. Are we clear? And then you have osmotic pressure that pulls fluid back into the capillary. So capillary osmotic pressure. Why there is no osmotic pressure from the capsular side? Why there is no osmotic pressure incurred by filtrate? There's no protein in the filtrate, right? It is not filtered into there. Please know what's going to happen to GFR if pressures change. Say, if blood pressure changes, which pressure changes? Hydrostatic, capillary. It increases. Are we clear? If I will tie down ureter, okay? filtrate will accumulate in the kidneys and capsular pressure will increase. There will be no drainage of filtrate. Does that make sense? If I will increase somehow protein concentration in the blood, then osmotic pressure will increase. I give you only the examples with increase, but you should be able to operate both ways. Are we clear? Okay? Now, if hydrostatic pressure from the capillary increases, what happens to the GFR? It goes up. If capsular pressure or capillary osmotic pressure increase, then GFR goes down. Does that make sense? Because those two pressures, capillary osmotic and capsular hydrostatic, they drive fluid back in the blood. Okay? I'm going to ask you questions about this. Okay? Right. Kind of capillaries fenestrated. What's so special about glomerular capillary bed? High blood pressure. And it's two arterioles on two sides. Podocytes, sorry. That wasn't my intention at all. Podocytes. Cells that surround capillaries, and food processes form filtration slits. Proteins and anything bigger than proteins cannot cross filtration slits. GFR, amount of filtrate formed per minute. 3 factors that determine it. Permeability of the membrane, which is super high and usually doesn't change. Surface area of the filtration membrane, which is usually high, sort of, but can be regulated, right, by mesangial cells. And then what I was talking about. And the pressures. Now, remember, the main way to regulate GFR is to change the blood flow, not surface area, not so much, blood flow. How do we change the blood flow? We dilate or constrict arterioles, right? Does that make sense? Physiological parameter defines net filtration pressure, blood pressure, basically. Okay, intrinsic controls. They maintain GFR. Extrinsic controls maintain blood pressure. Okay? So normally, intrinsic controls are important. Fluctuations in the blood pressure, kidney adjust for it. Does that make sense? What happens to GFR if blood pressure decreases? GFR decreases, okay? Now, if it decreases too much, what happens? What can happen to GFR if blood pressure drops, say, 30%? We have 3 pressures. We have hydrostatic pressure from the capillary, we have hydrostatic pressure from the capsule, and osmotic pressure from the capillary. If blood pressure decreases, which one is affected? Hydrostatic capillary. If it is decreased, what's going to happen to GFR? It's going to be 0. There is a point at which GFR is 0. When GFR is 0, what does that mean from the standpoint of kidney function? They don't work. They don't filter. So toxemia, right? They have toxins in the blood. Okay? 2 main mechanisms of the intrinsic control of GFR. I will ask you questions like that. Those are the most hated questions by you and by me. But I will ask them because I have no other way to ask. Remember, say, so those are intrinsic mechanisms. So myogenic, if blood pressure increases transiently, if it goes up, GFR goes up. In response, afferent arterioles will constrict, efferent dilate. Then operate this, work on this. Know how blood pressure change will change GFR. Kind of opening it up for you, I'm not going to ask about osmolality changes. Okay, so focus on blood pressure. Are we good? We clear? But there will be questions. So there will be questions about myogenic mechanism, and there will be questions about tubular-glomerular feedback mechanism. So this usually myogenic, it's just easy. Okay? Does that make sense? Tubular-glomerular mechanism. Do you want increased or decreased blood pressure? Decreased. Okay. When blood pressure goes down, how does it change GFR? Decreases. Follow with us because that's important now. So it decreases. When GFR decreases, filtration rate of filtrate formation decreases. Flow rate decreases. Reabsorption time increases. So when reabsorption time increases, more sodium chloride is reabsorbed, which means sodium chloride in the filtrate decreases. Which cells sense it? Macula densa. And what do they do in response? What we started with. Decrease in the blood pressure. So what do they need to do? Dilate afferent, constrict efferent. Here's the deal. The hard part about tubular-glomerular mechanism. We know what to do in the end. The hard part is how do we get there? Does that make sense? So I will ask you how we get there. There will be a question about it. I promise. Okay? Are we clear about it? Okay. Macula densa cells sense sodium chloride. Granular cells, they change diameter of the afferent and efferent arterioles and release renin. Remember, those are the sources of renin. Mesangial cells, communication. Structural features of mesangial cells. They have gap junctions. We just talked it through. This thing, that's the tubular-glomerular feedback mechanism. We just talked it through. Everything is recorded. Let's pray to the God of computers that it's not, halting during the process, but just let's keep our fingers crossed. And then it will be uploaded. All right? So this, what we just talked through. Are we clear? Extrinsic control. So what are two major mechanisms for the extrinsic control? Well, it's not two actually, it's more than that. In the situation with extrinsic control, if blood pressure, listen up, increases, what is the easiest mechanism to decrease it? When blood pressure goes up dramatically, what happens to the filtration rate? Goes up. What happens to amount of filtrate formed? Goes up. Blood volume? No. Blood, because we filter fluid out, blood volume goes down. That makes sense? Blood pressure goes down. Okay, we're more concerned with the low blood pressure. If blood pressure is low, what are the mechanisms? First, the direct mechanism that we just described. We just produce less filtrate. Does that make sense? Now, when blood pressure is low, which system is activated? Sympathetic system is activated. Okay? Now, sympathetic system is that's going to be cardiac stimulation. No, that's a different one. Sympathetic system, though, activation of sympathetic system will lead to the release of what? The enzyme from granular cells. Renin, which will activate angiotensin, which will lead to the release of aldosterone, which will increase reabsorption of sodium, which will draw water with it, which will increase blood volume. And there we go. Does that make sense? Okay. Another thing, sympathetic. Does that make sense? Now, angiotensin, which effect does it have on the blood vessels? Vasa? No. Angiotensin is vasoconstrictor, which means if afferent arterioles constrict, what happens to the filtration? It goes down, blood volume goes up. Sympathetic stimulation, overall, constricts blood vessels, filtration decreases, blood volume goes up. Does that make sense? So we got this: renin, angiotensin, aldosterone, epinephrine, norepinephrine will also constrict afferent arteriole, amount of urine goes down, blood volume and blood pressure increase. Renin, angiotensin, aldosterone. Just got it. GFR. So we got this again. Remember, when blood pressure goes down, macula, what happens is the rate of filtrate formation goes down, therefore reabsorption time increases because it flows slower, which means sodium chloride levels go down, and macula densa cells will send a signal to the granular cells to produce renin. To decrease blood pressure, it's that's what, adenosine and prostacyclin, and they vasoconstrictors. Okay, any questions about reabsorption? Just want to make sure that everybody clear on the directions. Annalise.
Adenosine and prostacyclin.
Prostacyclin. Those are prostacyclins, prostaglandins. They produced directly by kidneys in response to low blood pressure, and they vasoconstrictors. Sure. In reabsorption, few concepts that I want you to understand. So sodium-potassium pump. On the basolateral side, pump sodium from the tubular cells into the blood. We're clear. So sodium in the tubular cells is low because of that sodium-potassium pump. Okay, we're good? We're clear about this. So sodium in the tubular cells is low. So the consequences are, since sodium in the tubular cell is low, sodium diffuses via apical epithelial side of the tubular cells. Does that make sense? So this is the driving force, pumping out sodium on the basolateral side so that sodium can diffuse on the apical side. So all other, majority of other ions like calcium, chloride, glucose, amino acids, what is the process that is responsible for their reabsorption? Is that diffusion or the active transport? Active transport. What type? Primary, secondary? It's a secondary active transport because what is the driving force for that active transport? It's sodium diffusion. Does that make sense? And this secondary active transport happens on the apical side of the epithelium. We're clear? So secondary active transport driven by sodium diffusion on the apical side of epithelium. Paracellular, transcellular routes. Things that I want you to focus on. Paracellular, between, transcellular, through the cell. Remember? Paracellular route, between the cells. It doesn't cross any membrane. So paracellular route can be only what type of transport? Only diffusion. Because there are no transmembrane proteins, meaning it cannot be active transport. Which cellular structures do ions cross? Apical epithelium, cytoplasm, basolateral epithelium, which transport proteins. So, channels, seam porters, and just generally understand it's transport. Paracellular reabsorption, only diffusion. How is this gradient formed? Now, here's the deal. That's kind of, it's not tricky, but when sodium is reabsorbed, what is following sodium? Water. What happens when water leaves the filtrate? What happens to general concentration of filtrate? It increases, so all the ions increase their concentration, and that's the force for the paracellular reabsorption. Does that make sense? Transport maximum. Know this concept. I'm going to ask this. Okay? So think about this. You have a chemical. Listen up. You have a chemical that is filtered, and then it is usually, usually fully reabsorbed. So if the concentration of the chemical in the filtrate is under transport maximum, what's going to happen to the chemical? Fully reabsorbed. If it's above transport maximum, some will be excreted. It's going to be reabsorbed to that transport maximum, but whatever's on top will be excreted. Are we clear? Secretion. Physiological purpose of secretion is to get rid of a crap. Okay? No, seriously, it's the problem with the secretion is that mechanisms for secretion are very different and very specific for different ions. I'm asking about actually two. Urine and potassium. So potassium secretion is regulated by aldosterone. So elevated levels of aldosterone, how do they change potassium secretion? High aldosterone means increased potassium secretion. Are we clear? Urine disposal. Remember what happens when filtrate goes through all tubular systems? Water is being constantly reabsorbed, especially in the nephron loop. Does that make sense? So urea concentration should be urea actually. Urea concentration in the filtrate goes up. So at some point, urea starts to diffuse back into the blood. Does that make sense? However, one of the points of kidneys, why just 3 minutes digression. Kidneys of humans and kidneys of reptiles, well, humans, overall, all animals that can sweat. They're very, very different. Okay? Kidneys of reptiles, they employ completely different biochemistry that allows them to dispose of urea and uric acid without basically flushing kidneys. Why do we drink so much? We drink so much because we produce a lot of urea, and the only way to get rid of it is to flush it through the kidneys. We drink water to flush the toxins out. That's the goal. Nothing else. Now, that makes us depending on water sources, okay? But it also enables us to be warm-blooded, basically, okay? Reptiles, different story. You've heard the story of Peaches. I don't know if you've heard it. And Lizard escaped under my watch, and he was lost for 2 weeks, and he was just fine. He didn't drink for 2 weeks. Didn't have a sip of water or anything. And crawled out. And he's happy now again. Okay, so they have different water metabolism. Right? So that urea that accumulates in the filtrate will diffuse back in the blood, and it has to be secreted again to get rid of it, basically. Osmolality. Okay. So red is nephron loop, blue is vasorecta. And it's PCT. Let's overview it. Osmolality is highest. Where? The bottom. Now, the volume is highest. Where? Top at the PCD, right? Lowest here. So reabsorption in the nephron loop of juxtamedullary nephron is facultative or obligatory? Obligatory. We do not regulate it. We reabsorb all that water. Okay? So volume goes from the biggest here to the smallest here. Okay? Osmolality goes from small, huge, smallest. Okay? That's the most diluted point at the distal convoluted tubule. That's the most diluted filtrate that we have. Now, in the descending limb, what is reabsorbed? Water. In the ascending, sodium chloride. Know this. There's no logic. It's just a memorization. This part, okay? But it kind of helps to figure out what happens to the volumes. In terms of vasorecta, so this is, say, efferent arterial side, and this is venial side. So how does the volume of the blood changes in vasorecta as it approaches the vein? It increases because it picks up water and sodium chloride that comes from countercurrent multiplier, from the loop. Are we clear? Okay? So, countercurrent multiplier, what does it do? It reabsorbs the water and sodium chloride. Countercurrent exchanger picks up that stuff. So that's what we just discussed. What is osmolality? Am I going to ask you numbers? No, I'm not going to ask you numbers. You need to know the ratios. Am I clear? Ascending limb, so we got this. Osmolality of the filtrate at the end of the nephron loop. It's slightly lower than in the beginning. Slightly lower. Countercurrent exchanger, it's picking up water. Happens to the blood volume. We got this. Collecting duct. Which water-related process happens in the collecting duct? In the collecting duct, water can be reabsorbed. Now, is that facultative or obligatory? ADH. Remember, obligatory process is not generally regulated. I mean, obligatory means it happens no matter what. If the filtrate is produced, we're going to reabsorb the water in the nephron loop. In the collecting duct, now think about it. If you drink a lot of water, what happens to the osmolality of your blood? It goes down. ADH is produced? Nope. Because it's produced when you are dehydrated. Since you overhydrated, you have no ADH. No ADH means no aquaporins. No aquaporins means no reabsorption in the collecting duct. Means you produce, do you produce a lot of urine or a little? A lot. Is it diluted or concentrated? Diluted. Remember when we talked about ADH? Now, you have ADH. You dehydrated, severely dehydrated. So ADH starts to be released from the posterior pituitary. It increases number of aquaporins in the collecting duct. Blood starts to be reabsorbed. Okay, we're good. Boom. I'm just looking at it. Okay, yes. So what if if you dehydrated, because there's a lot of ADH, the volume of urine will be low and concentration will be high. Now, if you somehow inhibit ADH, you give some diuretics, the volume again is going to be high and the concentration is going to be low. Does that make sense? Urea, what you need to know, so you need to know that it maintains the medullary gradient. Does that make sense? Remember we talked about it, that the whole reabsorption thing in the nephron loop is because there is a gradient in the medulla. From the superficial medulla to the deep medulla, osmolality increases because of urea. Okay? So that's all I need, all I want you to know. So imagine this is medulla. So this is cortex. This is medulla. Urea concentration increases. That makes sense. Now, renal clearance. It's the parameter that shows whether a chemical is reabsorbed or secreted or additionally freely filtered. Let's go over it fairly quickly. If renal clearance is equivalent to GFR, what does that mean? Chemical is freely secreted. That makes sense. If it's 0, completely reabsorbed. If it's between 0 and GFR, partially reabsorbed. And if it's more than GFR, it is secreted. So it's filtered and secreted on top of that. Does that make sense? You're going to get a question about it. I'm going to ask you, such and such chemical is blah, 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 blah. Has such and such renal clearance. What does that mean? Okay? We're good. Okay, water loss, yeah. So we sweat, we breathe, we pee. It's all fairly similar. So, regulation of thirst. You feel thirsty. It's kind of a new thing for you here. To feel thirsty. Angiotensin makes you feel thirsty. ADH makes you feel thirsty. And then you compensate for the water loss by drinking water. Okay, now, water regulation. What you need to understand. We have two, except for the drinking water, we have two main mechanisms for the bringing water back into the body. It's ADH and renin-angiotensin-aldosterone system, right? ADH is purely water. Renin-angiotensin-aldosterone system, water follows sodium. We're good. Now, sodium, what you need to know. Sodium content is regulated, not sodium concentration. Sodium concentration is constant all the time. Why sodium concentration is constant? Because of the water. right? If sodium concentration goes up in any compartment, water will be attracted there. What I want you to know about sodium regulation. Renin, angiotensin, aldosterone system. Aldosterone, how does it affect sodium reabsorption? Elevated levels of aldosterone increase sodium reabsorption. Atrial natriuretic peptide, elevated levels of ANP increase sodium atrial natriuretic peptide. They increase sodium, usually say excretion, because it's mostly impaired reabsorption. Okay? Does that make sense? Do you understand what I'm saying? So, it may help you in English. The element is called sodium. But if you would look at the periodic table, the letters for it is Na. Because in the majority of other languages, it's not sodium, it's natrium. Okay? So natriuretic is the enzyme or hormone that passes sodium out. So elevated ANP increases sodium excretion. Stimulus for aldosterone, low blood pressure. Stimulus for atrial natriuretic peptide, high blood pressure. And remember, we talked about it. When you eat something salty, what happens to immediately happens to the sodium content in the ECF? It increases, which means since the concentration is the same, sodium content in the ECF increases. It draws it draws water, and volume of ECF increases, and you have a spike in blood pressure. Clear? Good? Potassium level regulation. For potassium, what I want you to know, two mechanisms. One is the direct excretion of potassium, just filtration, okay? And two, aldosterone-mediated secretion. Does it make sense? So direct filtration and aldosterone-mediated secretion. Why potassium levels have to be tightly regulated? Potassium is key for nerve transmission. If somebody develops hyperkalemia, atrial fibrillation. So main hormone regulating calcium reabsorption. Parathyroid. Period. That's all you need to know. Period. That's it. Know that PTH regulates calcium reabsorption. Three major systems. Know these three. So phosphate, bicarbonate, and protein systems, where they play the role. We all have it. Okay? Protein is in the blood, intracellular, bicarbonate, intracellular, extracellular fluid in the blood. Phosphate is in the urine. I'm not going to ask you about the chemistry of protein and phosphate buffers. Relax. Okay? Don't worry. For bicarbonate system, if you develop acidosis, acidosis means extra hydrogen ions. These extra hydrogen ions are going to bind to what? Bicarbonate ion. If you develop alkalosis, it means not enough hydrogen ions. Where are hydrogen ions? Along. These hydrogens are going to come from what? Carbonic acid. Okay? That I want you to know. We're clear. So, you know what I'm asking here. Please, like I tell you, extra hydrogen ion will bind to bicarbonate. Period. It actually doesn't matter. Even in the urine, it will bind to bicarbonate. Hydrogen ion will always bind to bicarbonate. So remember what happens during hydrogen excretion in urine. Remember, you have CO2 and water that form carbonic acid in the cell. In the cell, it dissociates carbonic acid. Hydrogen is going into the filtrate. Bicarbonate goes back in the blood. bicarbonate cannot be reabsorbed from the filtrate. Remember that. That's why we have to do this trick. So that hydrogen that goes into the filtrate will recombine with bicarbonate in the filtrate, form carbonic acid, which will dissociate into the CO2 and water. CO2 will diffuse back into the tubular cell, and the whole thing starts over. So that convoluted detour is for making bicarbonate that will go into the blood and hydrogen that goes into the filtrate. So that tool, just be able to recap it, okay? Acidosis. Let's focus on acidosis because alkalosis is the opposite. Respiratory acidosis. Why? Yeah, you're not ventilating well, okay? So in respiratory acidosis, what accumulates in the blood? CO2. What is the response from kidneys? They will try to excrete more hydrogens and to bring back more bicarbonate in the blood. So you will see elevated CO2 and elevated bicarbonate. In the metabolic acidosis, which one is decreased in the blood? Which ion? Decreased. In acidosis, bicarbonate is decreased. The response from respiratory system in metabolic acidosis. Breathing harder, which will affect CO2 how? Decrease CO2 concentration in the blood. You will get the question in whatever acidosis, alkalosis, metabolic respiratory. This is decreased, and this is the response from kidneys or lungs, okay? And then result of this response is that. Am I being clear? Basically, you saw that type of question on the quiz. Expect this something like that on the exam. Are we good? So we got this. Basically, if you answer each tiny little question here and here, it's the acid-base balance in the kidneys. It's that cycle that we just talked through. Respiratory alkalosis and acidosis and alkalosis. Respiratory lungs, can they compensate for respiratory? So one system has to compensate for another, right? That makes sense? Good. So this is more detailed. I will be honest with you. Focus on acidosis. Okay? Both respiratory and metabolic. Like, know what happens there, because you're going to see patients have restrictive pulmonary diseases or patients that have failing kidneys, but you're probably not going to see a bunch of patients that run around hyperventilating. Okay? I mean, they're going to pass out. That's it. And then they're going to come back and they're going to be normal. So, urinary bladder and urethra, they all lack submucosa. Remember that? There's smooth muscles in ureter and urinary bladder and urethra and epithelium. Epithelium, no epithelium. So it's transitional all the way to practically urethral orifice, where it turns into the stratified squamous. Okay? So know this epithelium things. Right? Trigone is the area between the orifices of ureters and the urethral like drainage. That's where the infections are. Male urethra is longer. What controls the... So, okay, efflux of urine. Micturition reflex. Bladder is full. It sends two signals. One is going to cortex. One it goes to the spinal cord. Okay? So one from the spinal cord, which sphincter is under autonomic control. Internal. External is under somatic control, right? So, in order to start urinating, parasympathetic stimulation increases, somatic stimulation decreases, relaxing the external urinary sphincter, and you pee. Does that make sense? Okay, that kind of know which system controls which sphincter. Am I clear here? System has to be inhibited. Yeah, sympathetic has to be inhibited. Parasympathetic has to be stimulated. Which part of the brainstem contains micturition center? Pons. Okay, we're good. No, I don't want to make a change. It's probably going to be one gigantic recording. Oh, crap. All right. Okay, now listen. If you don't mind anatomy, you can do anatomy. Can you? You can memorize. You don't need my help on the anatomy. The only pictures that you're going to see are the pictures on the PowerPoints. Okay? I'm going to hit the main points about physiology. This, physiological importance of ascending and descending, regulating sperm production. Okay? Maintaining the temperature when it's hot, it descends. When it's cold, scrotum ascends. Cryptorchidism is a condition when they do not, Cryptorchidism, it's when they do not descend in childhood, and they stay inside. Anatomical circumcision. Medical justification for the procedure decreases the risk of sexually transmitted diseases. Okay. Erection and ejaculation. Let's just go over the whole thing. So parasympathetic response. Parasympathetic ACH is released. It stimulates the release of nitric oxide. Nitric oxide dilates the blood vessels. Blood vessels deliver blood to the cavernous bodies and spongious bodies in the penis. Penis engorges with blood. Erection. Got it? For ejaculation, sympathetic stimulation does basically two things. One, it's the constriction of smooth muscle around the ejaculatory duct and vas deferens and epididymis, and ejection of the sperm. Okay? And the sphincter at the bottom of the bladder is closing, so there is no sperm going into the bladder. Okay, so parasympathetic is erection, sympathetic is ejaculation. Anatomical path for sperm. Seminiferous tubules, straight tubules, ready testis, epididymis, vas deferens, ejaculatory duct, urethra. Like, trace it. There will be a question for it. Am I clear? Okay. Oh. here. Two muscles that ascend and descend, dartus and cremaster. No. So if I ask something here, unless I strictly say don't do that, that can be an example. 3 accessory glands: seminal, prostate, bulbourethral. I'm not going to ask you. The seminal gland produces milky fluid, white fluid, no, yellow fluid, nothing like that. Okay? More of a functional thing: fructose, energy, alkaline component, neutralizing the acidity of the urine of the vaginal environment, clotting factors, holding sperm in the vagina, prostaglandins, stimulating vaginal contractions so sperm can be pulled in. Does that make sense? Physiological importance for this secretion: it's an activation of sperm and prostate-specific antigen. Bulbourethral gland produces mucus that lubricates glans penis and neutralizes traces of urine. Okay, we got this, and we got this. Don't focus on the muscles that propel. Smooth muscles propel the semen through the ejaculatory duct. That's as far as it goes. Again, know that it's sympathetic, and why the sphincter constricts. So sperm doesn't go in the bladder. Okay, sperm cell development. What I want you to know: start with diploid cell. Goes into mitosis. One of those 2 diploid cells is a spermatocyte. It goes through 2 meiotic divisions and produces 4 haploid spermatozoa. Am I clear? Okay. So basically, what I want you to do with this is to review the process. I will not ask you, is secondary spermatocyte, I don't know, has a tail or something like that? No. But just know that spermatozoa, the functional sperm cell, is haploid, and there are 4 of them produced from 1 primary spermatocyte. Are we clear? So they are produced in the seminiferous tubules. Sertoli cells direct the development and form the barrier between the blood and the testes, basically, so there is no exposure of the immune system doesn't develop autoimmune reaction to the sperm. Leydig cells produce testosterone, which is necessary for spermatogenesis. HPG axis. Just know them. Gonadotropin releasing hormone from hypothalamus stimulates follicle stimulating and luteinizing hormone from the pituitary. FSH stimulates androgen binding protein in Sertoli cells. Luteinizing hormone stimulates testosterone from Leydig cells. Testosterone binds to androgen binding protein, and that leads to successful development of the sperm. That makes sense. When sperm accumulates, which hormone is produced more and more? It's inhibin. When sperm counts rise, inhibin levels rise, and inhibin inhibits HPG axis. Testosterone inhibits HPG axis as well. Does that make sense? Now, when there are no gonadotropins or no gonadotropin releasing hormone, when those hypothalamic and pituitary hormones are not produced, what happens to testes? They start to shrink, right? And actually in people who, bodybuilders who abuse testosterone, that's really a common side effect. If they don't do it smart, then basically by taking testosterone, they inhibit gonadotropins and testes shrink. It's kind of paradoxical, sort of. Okay, metabolism. It's increased BMR, growth of everything, development of sex characteristics, oily skin, hair everywhere, growth of bones and increased bone density, muscle growth, deepening of voice. Just general topics. Female reproductive system. Yeah, the list that I told you. Just know them. Okay? Ovaries. That's where the follicles are developing. Fallopian tubes deliver follicle, mature oocyte throughout to the uterus and allow it to be fertilized. Uterus implantation of fertilized ovum. Vagina copulatory organ, type of mucosa, stratified squamous epithelium, fallopian tubes. Now that may be like, what? Ciliated. Epithelium in the fallopian tubes is ciliated to propel the ova. Two layers found in ovaries, cortex and medulla. In cortex, there's actually follicular development. In the medulla, there are blood vessels and nerves. Ovaries and fallopian tubes are not physically connected. What happens if oocyte ends up in the abdominal cavity? Nothing. I mean, it dies. Ectopic pregnancies, the pregnancies in the abdominal cavity or in the fallopian tube. Risk factors, sexually transmitted diseases, pelvic inflammatory disease, infection that crawled through the fallopian tubes and ended up in the pelvic cavity, sexually transmitted diseases, long-term negative outcomes, infertility. 3 layers of endometrial wall. Perimetrium, which is just connected. Myometrium, muscle, okay, contractions of the uterus. Endometrium, 2 layers, stratum basalis and stratum functionalis. Stratum functionalis is where implantation happens. Stratum basalis allows the growth of new stratum functionalis. Scrotum, bulbourethral gland, penis, glans penis, well, prepuce. So, just, do you understand what I'm asking here? So, labia majora develops from the same structure from which scrotum develops. Glans penis is essentially the same part as, glans penis is the same part of glans clitoris. Prepuce of the clitoris is the prepuce of penis. Does that make sense? So know this. Modified sweat glands, ducts, and then sinuses. Now, major source of cancer cells. Breast cancer is epithelial cancer. Okay, risk factors, late menopause, no late pregnancies, no breastfeeding, genetic mutations, BRCA1 and 2. Just know them. Probably not going to ask about it. You know them already. Just for your sake. Ogonium diploid or haploid. It's diploid. When it divides, it produces primary oocyte. It's diploid, and then a primary oocyte enters the meiosis. Does it complete it? No. It completes it during puberty, right? Sorry, the brain freeze now. Yeah. Remember during puberty, every month primary oocyte completes meiosis 1, enters meiosis 2, and stops there, is the secondary oocyte. Okay? And secondary oocyte is the part of the mature vesicular follicle. So the thing that I want you to know is that secondary oocyte, which ovulates, is the part of vesicular follicle. Secondary oocyte, does it complete meiosis 2? It does not. It still has to complete it. It's in the prophase. Prophase? Metaphase. I'm not going to ask you which phase. So, meiosis 2 is completed by oocyte after it's fertilized. We're clear. It's fertilized. It completes meiosis 2, and it becomes ovum, fertilized ovum. Now, from one oogonium in the end. So how many secondary oocytes are produced? How many ova are produced? 1. We have 3 polar bodies and 1 ovum. Does that make sense? Now, spermatogonia produces 4 sperm cells. Remember, we talked about that difference. Maybe physiological importance of it. It's really hard to raise 4 babies and to carry 4 embryos and then 4 fetuses. Okay, so follicular stage and ovarian, follicular stage and luteal stage. Things that you need to know. So, follicular stage, follicle develops. How deep should you know that? That's basically it. It is growing, becomes vesicular follicle, ovulates. After ovulation, corpus luteum is the remnant of the follicle, and it produces progesterone and estrogen that will help to support uterus in case there's fertilization. Am I clear? Okay. Hormone levels. During follicular stage, remember, estrogen and, not estrogen, FSH and LH are fairly low because estrogen inhibits them, right? But estrogen levels, they rise because follicle grows. That makes sense. And as it grows, it produces more and more estrogen. Okay? Which hormone stimulates follicular growth? FSH. Which molecule stimulates estrogen? LH. Does that make sense? When the levels of... So basically, product of the granulosa cells are estrogens. Okay, so Tickle cells produce androgens, and then androgens stimulate granulosa cells produce estrogens. I'm not asking that. I want you to conceptualize it rather than memorize all those cells. So, again, follicular phase, LH and FSH low because they are inhibited by estrogen. But estrogen is produced by the growing follicle, and the bigger the follicle, the more estrogen is produced. Are we clear? At some point, estrogen goes to the level where estrogen and luteinizing hormone establish positive feedback. That makes sense? There is a surge. So what happens? More estrogen means more LH. More LH means more estrogen, means more LH. That surge leads to what? Ovulation. What happens to levels of estrogen after the ovulation? They go down a little and then they go up. Progesterone starts to climb after the ovulation, because what produces progesterone? Corpus luteum. So during luteal phase, progesterone and estrogen are up. We're clear. Now, progesterone is up, estrogen is up, inhibin's up. Therefore, gonadotropins, LH and FSH, are down. If there is no fertilization, what happens to corpus luteum? It degrades. As it degrades, estrogen goes down, progesterone goes down, and HPG axis is not inhibited anymore. Does that make sense? So the whole thing starts over. So if secondary oocyte not fertilized, levels of inhibin, estrogen, and progesterone. How does it change? So when corpus luteum is degraded, these three go down, GnRH increases, gonadotropins increase, the whole cycle starts. I outlined it here. That picture, that picture, that graph for hormones, know it. I'm going to ask. Okay? Menstrual phase. Be able too much. So menstrual phase. Blood vessels constrict, deprive stratum functionalis of the blood flow. It dies. Rush of blood washes it away. Are we good? Levels are low. Mechanism for shedding. Proliferative phase. Which hormone is elevated during proliferative phase? It's estrogen. Okay? Progesterone is still low. Proliferative phase, when new stratum functionalis is formed. Secretory phase. That's progesterone. Progesterone increases, progesterone stimulates stratum functionalis to be able, I lost my words. Enables it for the successful implantation of blastocyst. Okay? Levels of estrogen and progesterone are high and they become low if the oocyte is not fertilized. Growth up, breast development up, subcutaneous fat up, calcium balance. Calcium is deposited in the bones, blood cholesterol is low. That just review this table. Fertilization in the fallopian tubes. Acrosomal enzymes. They break down zona pellucida. They break down corona radiata. They break down corona radiata. Sperm cell enters. And then remember, there's a huge calcium surge that closes the door behind that sperm cell. So there is no polyspermy. Okay? DNA in sperm cell and ovum, they fuse. Fertilized egg is transported via fallopian tubes with cilia. Beat it. Blastocyst implants into the endometrium. Trophoblasts, they eat the cavity for the blastocyst and provide feeding in the first, basically, less than trimester of pregnancy. Then placenta takes it over. So, uterine arteries take the blood to placenta, uterine veins from. There are sinuses around placental villi. Okay? So, those sinuses are maternal. In the villi, they are fetal. So, umbilical vein carries the blood from placenta to the fetus. Umbilical artery carries the blood from fetus to placenta. Which blood vessel carries oxygenated blood? Umbilical vein or umbilical artery? In this case, to the fetus, which means vein, right? So umbilical vein will carry oxygenated blood from placenta to the fetus. Double bore effect. So CO2 goes from fetal blood to maternal blood. pH in the fetal blood goes up, which increases oxygen binding. Joseph, how about we review the quiz? 3 minutes, and so I know that you need to. No, it's okay. I just thought that I will review the quiz with everybody, and we're actually going to record it then. Okay. So quiz review. Can we do it now? So for Bohr effect, pH in the fetal blood goes up, in the maternal blood goes down. As pH in the maternal blood goes down, oxygen unloads and gets in the fetal blood where pH is up and it binds to hemoglobin. So that's double Bohr effect. And also fetal hemoglobin has higher affinity to oxygen. Healthy endometrium, stops menstruation, maintains corpus luteum, elevates progesterone and estrogen. So estrogen increases sizes of uterus, breast, and genitalia. Progesterone decreases uterine contractility, prepares breast for lactation, maintains healthy endometrium. Somatomamotropin, all you need to know, it stimulates the breast development. That's it. Preeclampsia, fluid accumulation, high blood pressure, kidneys are slow shutting down, which leads to toxemia. And also arteries start to constrict. Eclampsia is preeclampsia taken to the next level. So basically, severe toxemia, blood pressure through the roof. Which ovarian hormone initiates contractions in labor? It's estrogen. Okay? Remember, there's estrogen and progesterone. Progesterone prevents contractions. At the end of pregnancy, it goes down. Contractions are stimulated. Contraction, mechanical stimulation of the cervix, signal to the posterior pituitary, oxytocin. Good? Oxytocin leads to the increased contractions, more pressure on the cervix, more oxytocin, more contractions, more pressure. Milk production, what I want you to know. Suckling on the nipple stimulates two separate things. One is milk production. So prolactin release factors from hypothalamus stimulate prolactin from pituitary. Prolactin stimulates milk. Oxytocin stimulates contraction of the ductals and ejection. Do I make sense? Oxytocin has nothing to do with milk synthesis. Are we clear? Questions?